Okay, so good afternoon. Welcome to week 5 live interaction sessions for the immunology course. As you're already familiar, we will be taking up some of the topics that were already discussed in the past week. I will be addressing a doubt that was brought up in the previous class. And also, we'll be looking at the assignment that was discussed, that was given as part of the coursework in the previous week, and go ahead with it. If there is any doubt, you can ask it at any point of time. And of course, you can refer to the sessions online as are already available on YouTube or from the Google Drive link. Now, before I get started with this week's course, something that was asked about in the last session, I had addressed it. Just give me a second. Okay, so I can't exactly find that slide right now, but basically, people were some one of the students had asked about the fact that which of the antibodies are actually able to cross the placenta, and there was some confusion regarding it. So I had gone back to the multiple papers, publications, and research work that is done, and I had double checked and verified that indeed IgG is the only antibody isotype that is known to cross the placenta. And even among IgG, different subtypes of IgG have different frequencies or propensities to cross the placental barrier, or transplacental migration is unequal among the different subtypes among the IgG isotype. And in the slides that I had uploaded, these are actually the previous slides, so it's not updated here. But the ones that I have uploaded on Google Drive, you will find the link also for the paper given, so you can check that for your reference. So just to bypass that confusion, I thought I'll address that first. Now, coming to the week 5 discussion. So, first of all, let's go over the assignment questions for the previous week. Most of them were quite straightforward and were related to the topics that we had previously discussed in the session, or it was already taken up in the week lectures by Professor Shudeep. So the first question was about which of the following immunoglobulins are derived from the same pre-mRNA transcript. So if you remember, we had discussed in detail about how isotype switching is carried out, what is the arrangement that is there. So we saw that IgD and IgM, or otherwise indicated as mu and delta, they are the first isotypes, and they are synthesized from the same pre-mRNA transcript. And then, depending upon the type of pathogen that has invaded into the system, isotype switching occurs, and therefore different types of immunoglobulins can take over. That therefore the first question's answer is IgM and IgD. Second question was about class switching, and while the immediate answer obviously goes towards cytokine signals, because we had discussed how cytokines are actually responsible for carrying out that switch from one isotype to another. But if you go through the options carefully, you will see that switch regions, switch recombinase, all of these have also been indicated, and in fact they are. Yeah. So, in fact, they are necessary. Cytokines are only able to deliver the signal that isotype switching is required, and they're able to inform that from IgM, IgG, or specifically say IgG2 needs to be brought about. But the execution is carried out by the switch regions and switch recombinases, which is something we had discussed with the previous class, that the gene segment rearrangement will be coordinated by the trans factors and the cis elements. In this case, the cis element is the switch region, which is a uh, part of the chromosome or part of the gene segments. But switch recombinase is the enzyme, and therefore it is the trans factor which comes and sits on the switch regions and mediates this gene segment rearrangement, and therefore allows class switching to actually occur. Um, therefore, the correct answer in this case would be all of these. Then in question 3, we were asked about the first class of immunoglobulins that are produced after activation of a B cell is IgG, IgM, IgA, or IgE. So again, because IgM and IgD, like we discussed at the first subtypes, among them IgM is would be the correct option in this case based on the other alternatives that we have. Hopefully there was no confusion there. They were very straightforward questions. If there is, please let me know. Then comes junctional diversity. So we had discussed this as well as a type of adding to the existing variety in antibodies, or enriching the antibody diversity. So in this case, junctional diversity refers to which of the following phenomena. Now, these are actually quite similar, but not exactly the same, especially option A and option C can seem very similar. But again, you will see the correct answer has obviously been indicated, and you'll see the difference between them as well. So, junctional diversity refers to A, random addition or deletion of nucleotides between junctions of immunoglobulin gene segments. That is definitely the correct answer. And we had looked at it that depending upon where two segments or where the recombination is occurring between two segments, depending on that, the diversity can vary, and it will enrich the antibody variety that exists, or can lead to multiple combinations, of which some would be productive rearrangements and some would be non-productive, and only the productive ones would be allowed to proceed forward and would eventually lead to generation of antibodies that are creating a repertoire with higher diversity. The non-productive rearrangements will not be allowed to go forward and would be eliminated from the system. Second was availability of multiple copies of J chain segment. That is not a junctional diversity. It is because of the it can actually be considered as part of the combinatorial association of heavy and light chains, because or maybe not that also. It is actually the recombination between different segments, and how VBJ recombination is occurring. It adds to diversity in that sense. The 1223 rule of segment joining is also similar, but is not the same. It is not actually junctional diversity in this case. While 1223 rule ensures that there is an orientation that is maintained, that whether or not it will be it will be a signal joint that is getting excised out of the genes, or out of the gene segments, or whether it will lead to deletional joining or inversional joining. All of that is determined based on how the orientation of the 1223 regions has been arranged. So that actually forms a part of how gene segment rearrangement is occurring, and how VDJ recombination can take place. It is not a part of junctional diversity directly. Junctional diversity is just between different junctions, how nucleotides can be added or removed. And under junctional diversity, we had also looked at P nucleotide and N nucleotide addition and how they are executed. So option A is the correct answer in this case. The reason I'm elaborating on this more is because many a times it's easy to get confused between option A and C, but you need to keep your head clear about this, that 1223 rule is for the type of joining that will take place, whether or not it will be an inversional or a deletional joining. That is what this particular phenomena determines, whereas junctional diversity is different. So I hope it was clear. If not, you can go back to the slides, or you can ask me at any point of time. We have clear schemas for each and every subtype of these ways of getting more and more antibody diversity. Then comes the next question. Dimers of IgA are formed by A, disulfide bonds, B, alternative splicing, joining by a J chain, or joining through hinge region. So we have not really discussed the exact roles of each of these antibody types in a lot of detail, but we had schematics for each of these. If you remember, we had elaborated on it several times, but I'll just go back to it quickly. Yeah. So here you see the IgA dimer, the way in which it has been schematically represented. It has a J chain, which is joining these two, shown here in blue. And in case of the IgM pentamer also, we see that J chain is present there as well. But the pentamer structure cannot be formed only by the J chain, but instead it also needs to have multiple disulfide bonds linking it. Only then that entire elaborate structure or pentameric structure can be formed. So there's a difference between these two. So just wanted to highlight it, because the options again can seem very confusing sometimes. But yeah, in case of IgA, it's actually J chain, which maintains the dimerized state of IgA. Then comes T cells use all of the following for generating antigen recognition diversity on the TCR, except which of the following. So all of the options that you see here are true for BCRs or antibody diversity, but in case of TCR or T cell diversity, one of them is not true, and that is somatic hypermutation. There is combinatorial association of chains that is heavy. That is for the TCR. The chain association is different. The association of segments or gene segment rearrangement is also occurring, there is nucleotide addition which takes place, but somatic hypermutation does not occur. I don't think I have elaborated directly on T cell diversity or TCR diversity generation, but it was there in the lectures and it has been taken up in one of the separate videos, if I'm not wrong. So you can look back at that, or if necessary, you can look at Kubi for your reference as well. And also, I now have the PDF for the 7th edition of Kubi Immunology. So maybe I'll upload that in the Drive link, so you can access that if you want. Most of my previous schematics were from the 5th edition, so they were slightly backdated. Not slightly, they were quite backdated. So I got the more updated version. I still don't have the current edition. I think it's the 9th or 10th edition. The PDF is not available, but the 7th edition also has a lot of added information. So I'll maybe share that for your for easier reference. So yeah, this was the question that was taken up. Then we were asked about B cells undergoing immunoglobulin class switching. So class switching, what exactly is changing after class switching has been executed? So whether the variable region changes or the constant region changes is how you'll eliminate options. So obviously we know that when there is class switching, it is the C region, which is the fragment crystallizable, that is actually changing, and that is giving rise to the IG, G, A, M, E, and D. And therefore constant region must change. So option B and C obviously become our preferences. Among them, you have to see the variable region for which of the two remains the same. It is actually addressing only the variable region of the heavy chain, because class switching will only occur after the antibody has seen its cognate antigen and that B cell has undergone activation or clonal selection. And therefore, it is now decided that to tackle whatever antigen has come in, you need to have the same variable region, but you need to change the constant region to tackle that pathogen more efficiently. So in this case, option C would be correct. Again, these options are very similar. You eliminate options based on what knowledge you already have, and that should help you, figure out or troubleshoot how exactly you can address questions like this. So keep in mind the variable region of the light chain also remains the same. You are not changing that. So in this case, somebody might ask that why is option B not the correct? The variable region is not affected. But in this case, when you talk about class switching as a phenomenon in particular, then what becomes more important is that the heavy chain is what undergoes changes, and the heavy chain is what participates in the phenomenon of class switching and not the light chain. So while statement B is also true, statement C actually addresses what is happening during the phenomenon of class switching. So that's how you need to tackle the question. If in case we were asked about which of the following statements are true, then in that case, both B and C would have been correct. But yeah, that's not the type of question asked. So you need to keep in mind things like this also. And especially if you are appearing for, exams like NET or something, if you're hoping to get into research maybe eventually, then there are sometimes questions where you get multiple correct options. You can have multiple correct options, let's put it that way. So in that case, you need to be a little more careful. But here we have a single question that is being asked, and it's more specific about the phenomenon. So in that case, option C is correct. Yeah. Then comes the mechanism that permits immunoglobulins to be synthesized in either a membrane-bound or a secreted form is A, allelic exclusion, B, class switching, the one-turn, two-turn joining rule, or differential RNA processing. So first of all, the one-turn, two-turn joining rule is the same as the 1223 rule. So that's why I used a schematic from a different book, because if you only refer to one book, then the one which was discussed by Professor Shudeep, then there it was mentioned as 1223. But if you remember the diagrams that I had shown in my slides, there we had one turn and two turn RSS, which is for recognition signal sequence, and that actually determined the inversional or deletional joining. But yeah, that is not what we are asking here. Here we've been asked about membrane-bound or secreted antibodies. So the way in which, not antibodies, immunoglobulins, let's put it that way. So that is a result of differential RNA processing. So if you remember what we had taken up as the first topic for the previous class. So we had talked about all the different types of PTMs that can occur. And I had specifically highlighted that it's not always necessary that alternative splicing should directly be the answer, because alternative splicing is of many different types. And I had shown all these different subtypes and what they're called: mutually exclusive exons, constitutive splicing, alternative 5' splice sites, etc. All these different varieties do exist. But in case of a secreted or membrane-bound form of antibodies, it was actually the way in which the RNA was getting processed, whether or not it carried the secreted or secretory region, or it removed the same, determined whether the antibody would remain membrane-bound or it would be secretory in nature, whether it could go into circulation. So this is what we had seen previously. So it's actually the event of differential RNA processing, and that processing can take place in the form of either splicing, differential or different ways of splicing, or it can take the form of polyadenylation as well. Especially here, it was specifically polyadenylation that had taken over the decision of whether or not it will be a secreted or a membrane-bound antibody. So differential RNA processing in this case is the correct option. So in case you had alternative splicing and differential RNA processing as the options here, even then differential RNA processing would be the correct option, because polyadenylation actually predominates over it. You do need splicing to be carried out in a certain manner. But yeah, for specifically generating the secreted form of an antibody, polyadenylation takes up the major role. So that is the correct option. So I hope that is clear. But yeah, you can obviously refer to the schematics that have previously been discussed for your own clarity. Uh, the next question that we have not directly taken up, but is definitely relevant, was about foodborne pathogens and what sort of antibodies they actually encounter. So while we did not take it up directly, I think we had this comparison between all these different sort of antibodies that have been shown. And if you look here, mucosal transport has been addressed. So mucosal transport is taken care of by these antibodies mainly, which are IgG-A1, A2, and IgM. Specifically among them, IgA1 and IgA2 are shown to be quite reactive towards mucosal transport. Why is this relevant? Is because when we talk about a foodborne pathogen, then that is obviously something that is passing through your gut. Something that you have consumed is passing through your gut, and specifically the intestinal lining. And the intestinal lining has the, if you remember in our very initial sessions, I think for week 1 or week 2, when we were discussing secondary lymphoid organs and secondary lymphoid tissues, we had talked about GALT and MALT. So GALT was gut-associated lymphoid tissue and MALT was mucosa-associated lymphoid tissue. So that is the immune system that resides within the gut lining, which is within the intestinal lining. And therefore, the subtype of antibodies which are able to cross the mucosa will be the first line of action that exists to counter any pathogens that come in with your food. So something you consume, if it has some sort of, say, a parasite, a helminth, you consume for whatever reason, consume dirty water from somewhere, or, some roadside food or something like that. So in that case, before all of the other subtypes of antibodies can take over, it is actually the antibodies present within the gut, which will be the first line of action, and therefore IgG-A takes over. Here we also see IgM, but its propensity is much less. And if you refer to, current research papers or even updated books, this also was from the older edition of Kubi. But if you refer to current literature, it says that IgA is the subtype that takes over this job of tackling foodborne pathogens. So that is the correct option. That's why I said that while we did not directly, bring this up as a topic, that IgA is specifically involved in this job, or IgE is specifically involved in this job, you can obviously refer to this table and you'll see that each of them have different functions. For example, if you're asked about, eliciting a allergic reaction, then of course the answer would be IgE, because it induces mast cell degranulation. So we will take up these during our hypersensitivity classes, which are towards the fag end of the course. But just for your reference, you can see that when you look at comparative tables, you have to figure out the conclusion from these tables that exist. So I hope it is understandable now. So the correct option in this case was IgA, because a foodborne pathogen will first encounter it. Then for the last question, we were asked about MHCs. So we had just touched upon the idea of MHCs and the basic structure of MHCs. We will be discussing it in a lot more detail today, and it has been discussed by Professor also during the course lectures. So which class of MHC molecules are able to bind to smaller peptides with 8 to 10 amino acids? So we had talked about the way in which peptides are able to bind to the MHC molecules. And of course, the correct answer in this case was MHC1, which was able to bind to smaller peptides or shorter peptides, versus MHC2, which had longer peptides. So in case you just need a quick revision. So yeah, this was class 1 MHC representation and class 2 MHC representation. You see that this has 8 to 10 amino acids as the average length, and MHC2 was able to bind to much longer peptides. And the way in which it was bound was also different. It had a certain orientation via which it could bind, and you can see that it was only the alpha chain, alpha 1 and alpha 2. From this structure, alpha 1 and alpha 2, which were responsible for forming the peptide binding cleft. Whereas for MHC2, it is alpha 1 as well as the second chain also participated, which is beta 1 also participated and generated the peptide binding cleft for MHC class 2. So we have already discussed this, so I'm not reiterating that. So in this case, smaller peptides are bound by MHC1, which completes our assignment questions for the previous week. I hope it was clear. If there are any doubts, please interrupt me at any point of time. And you can obviously refer to the slides at any point of time. They are most of the topics that have been discussed. They are covering what has been discussed in the assignments with additional information, some experimental data. Also, I try to include experiments that led to those conclusions because I feel it makes it more relevant to somebody, at least somebody who's interested in research. I'll be doing that today as well about MHCs as well. So we'll come to that very soon. Please let me know at any point of time if I'm going very fast or if something is not clear. I'll try to repeat it again. And if any queries, please do interrupt. So having said that, we'll start our discussion for today's session. So for major histocompatibility complex or MHCs, we have already looked at this figure before. This was about when I had introduced how MHCs are arranged within the let's say the genome of mice versus humans. So mice has been given here as reference because, like I said, most of the research that is done, whether it comes to disease biology, immunology, or for studying any human disease, obviously we always prefer to look at some sort of animal model, and mice are the most common animal models. There are also Drosophila, which are used for neuronal diseases. Fish are used for a number of diseases for studying regeneration, in some cases cancer as well. Different animal models are chosen. Mice in this case are most relevant because their immune system is very, very comparable to ours. And therefore, what we can, whatever we want to study can initially be, a preliminary or a preclinical study can be done in mice. Having said that, there are different types of mice that are commercially available. I don't know if you're already familiar with it, but most mice that are usually used for these immune studies, immunology-based studies, they are all called as immunocompetent mice, which means they have a functional immune system. But there are also several variants of these mice that have different immunological backgrounds, which means that they carry certain mutations which have been artificially induced by humans to generate some immunocompromised mice. Now, the question could be that why at all do we need immunocompromised mice? Sorry. Why do we need immunocompromised mice? So the main reason why it comes in is because for cancer biology, it is very relevant whenever we want to implant or grow a tumor in mice. For that purpose, you need to have an immunocompromised system. Why do you need that? So, for example, if you want to check, suppose a drug X has been discovered and you are claiming that it cures breast cancer, for example. So how would you prove that? You cannot directly give it to a patient and start a clinical trial. The first round of studies are carried out in mice, where mice are induced to carry those cancerous cells. Female mice would obviously be taken up and they would carry some breast cancer cells. And when you give that drug treatment, you will check whether that tumor shrinks without killing the mouse. What is the dosage that works? How does it work? Whether it has any, off-target effects, etc. So for that purpose, the type of mice that are used are called as immunocompromised, which means they carry certain mutations. Either their B cells and T cells are not working, or their NK cells are also not working. So there are different subtypes. If it is interesting to you, you can go and read up on it because it is used for disease modeling as well, and it is extensively used. I use it in my research. I work on cancer biology, so we work on immunocompromised mice regularly. They're called as not skid mice or skid mice for that purpose. So you can read up on it. It's very interesting. Uh, Now, having said that, we come back to again our topic at hand. So this is the arrangement of the MHC locus in mice versus the one which is present for humans. So I had briefly discussed all the different types. I mentioned MHC class 1, 2, and 3, and what are those regions and what do they encode as the gene products for your reference. Today, we'll be taking that up in more detail. So specifically, let's look at the human HLA complex, which was the human leukocyte antigen, or the MHC complex for humans specifically. So here you see a schematic representation of the genomic arrangement of the same region. So like I said, this part is present in Kobe Immunology 7th edition. So you'll find a lot more updates in the current edition, and you can refer to that. So for humans, the arrangement of these all these MHC genes is at chromosome 6. So again, this can be a, at any point of time, this can be a question also that is useful. Where are all these MHC genes located? So for humans, it is chromosome 6. So there you see the entire complex or the entire MHC locus resides within 4000 kb. That means 4000 kilobases, which means 4000 into 10 to the power 3 bases. So that's a huge region over which it is spread out. Within that, you see that if we look at the arrangement, first you have MHC class 2, which is shown here in blue, then in MHC class 3, and then finally MHC class 1, which is the same as is shown in the tabulated format. Within that stretch, you see that there are different gene segments. What you need to keep in mind is that when we are saying that it is MHC class 2, it's not just these DP, DQ, and DR genes that are there. There are also several other genes which you can see here indicated in black, which correspond for other factors that play a huge role in the entire antigen presentation and MHC function process. What are those? You're already familiar with them if you have listened to the lectures. So you have LMP2, LMP7, you have TAP1 and TAP2, and you have TNF alpha and lymphotoxin A, or rather lymphotoxin alpha. So LMP2, LMP7 are proteasome-like subunits. We'll come to that. Then we have TAP1 and TAP2, which are part of peptide transporter subunits. You might already be familiar with TAP proteins and what they do, but we will be taking it up later on. And you also have TNF alpha and lymphotoxin A, which is for tumor necrosis factor alpha and lymphotoxin. So these are all part of the MHC complex, and that's how the arrangement can be seen here. Following that, within the MHC class 3 region, what you see as a major stretch are for complement proteins, which is indicated as just a C dash. Like it was mentioned in the lecture also, we will be discussing complement in details later. It's an entire cascade. It's a very elaborate cascade for that matter. We'll be taking it up soon. And apart from that, you have TNF alpha and lymphotoxin alpha here also, which is part of MHC class 3. Then within MHC class 1, we have B, C, and A regions, among which you see the arrangement of HLA B, C, A. All of them are present in different regions, and this is the entire location that you can see. So also what you can see as an arrangement is that this is the centromere and this is the telomere. So it shows you the orientation of the chromosome. So in case I hope you're already familiar with it, but that's how the chromosomal arrangement occurs. So centromere means, okay, centromere actually indicates the center of the chromosome. I can show you a quick, just a second. Yeah. So if we look at if we take this representation of a chromosome, then this part at the center will have the centromere and the ends of the chromosomes will carry the telomere. So between these regions is the arrangement, not just this end, it can be this end also for that matter. That will be the telomere, which is the end of the linear chromosome. And at the center, you have the centromere, and within these regions lies the entire MHC locus spread out over a region of approximately 4000 kilobases. So that is the reference you can, like the arrangement that you can keep in mind. Now, having said that, I'm issue with the cursor. Yeah. So, having said that, we now move into more details about the MHC complex. So, first we learned about the structure, the need for each of these different subtypes, the type of antigenic peptides they're able to bind, and what exactly is the sub-arrangement within those major regions. We looked into all of that. Now we learn about more details about MHCs. So, one of the first most important things about MHCs that was discovered was the fact that how is MHC inherited? That where are they coming from? If you're saying they're part of the genomic locus specifically within this region, and this is the arrangement, and so on and so forth, the question is where are we getting them? For example, when we talked about antibodies, we learned that there are VDJ regions, and there is an insane amount of diversity that can be generated in the orders of 10 to the power 5, 10 to the power 7, and all of the different all of these different subtypes of antibodies can be brought about because of 5, 6, or 7 different factors. So we learned about all of that. When we come to MHCs, their diversity is limited and is limited for a different reason. So we'll come to that. First of all, we are saying that allelic forms of MHCs are inherited in some linked groups called as haplotypes. So entire MHC is inherited like from parents. MHC gets inherited directly, and that inheritance occurs as a chunk or as a haplotype. So what does it mean? It means that the genes that reside within the MHC region are highly polymorphic. What does that mean? It means many alternative forms of each gene or each allele exists within the population. Now let's elaborate that a little bit. On one hand, we are saying that MHC does not have that huge diversity range as much as that of antibodies, but we are saying that it is polymorphic. What does that mean? Polymorphic means that when you take a population into account, say for example, if we take the population of India, then at large, within the entire Indian population for MHC1, you will find that within MHC1, within one specific locus, say for example HLA-A, that locus is very variable. That while MHC by itself is not generating a lot of combinatorial diversity, but that locus can carry a lot of difference between one individual and another. For example, if I take my MHC locus and my parents, that might be quite similar. But if I take that of mine and that of any strangers, if somebody from my workplace or somebody from my school that I have no direct blood relation with, then in that case, our MHCs can be extremely different from each other. While they would have some regions which are constant, there is huge variation in certain regions, and that actually generates all the diversity. So that means that within a population, when you take a large set of people, then within that population, you will find that there can be a huge polymorphism or huge variation for MHC locus. Most individuals inherit all the alleles that are encoded by these genes as a set. So its inheritance is not occurring as a result of a large amount of recombination, but instead it is inherited as an entire chunk. So from the parental chromosome, that entire region is taken as it is, and that occurs because of a phenomenon known as linkage disequilibrium. What does that mean? It means that because some regions are so closely located with respect to each other that recombination cannot occur between them. Let's go back to this figure again. So you saw that these huge number of genes are localized within 4000 kb. If they were very far apart from each other, if say for example, let's take this chromosome as reference. If they were located, say for example, one set of genes was here and another set of genes was on another whole other chromosome, then in that case, recombination or the propensity of recombination or the probability of recombination would be much higher. But instead, what occurs, they are localized within a limited area within one chromosome, and therefore within that chromosome, intrachromosomal recombination, or rather because of the fact that they are so closely linked next to each other, the probability or um, the propensity of that region to undergo recombination is very, very less. And that is otherwise known as linkage. So when genes are very closely linked to each other, then they are inherited as an entire set. They do not undergo recombination, and that leads to disequilibrium. So that phenomenon is known as linkage disequilibrium. If there was no linkage, then there could be recombination. But because of the presence of linkage, there is a disequilibrium, and therefore no recombination can occur. I hope it was clear. I repeated it multiple times, but yeah, this is the phenomenon. This is how certain loci within our entire genome are always inherited as a set, whereas other regions have a higher propensity of recombination. There's actually like a formula, mathematical formula to calculate this. I had not included it directly. You'll find that in Kubi within like a separate like study box where you'll be able to see what is the recombination, how to calculate the recombination frequency. So you can do that quite easily based on how far apart they are. But MHCs do not undergo this phenomenon, and therefore they are considered to be linked genes, or rather linked alleles. So this set of linked alleles is referred to as an haplotype. An individual inherits one haplotype from the mother, one haplotype from the father, and effectively gets two sets of alleles and therefore is a diploid organism. So now we'll come to the experiments that actually showed how this was discovered, and hopefully that will clear it up a little bit. So here are some definitions that you just need to keep in mind. What is polymorphism, or why is MHC region considered to be polymorphic? Secondly, what is linkage disequilibrium? And thirdly, what is a haplotype? So most of these definitions I've explained here, but yeah, of course, if something is not clear, we can take it up. Let's go through the experimental basis of how this discovery was done first. So the illustration of yeah. Yeah, so we were talking about the experiment. So how at all can it be studied? So this will show you the experiment that was done to understand how MHCs were inherited, and for this purpose they use inbred mouse strains. What does that mean? It means these are mice that are part of the same colony and they have been breeding among each other. Not they're not Okay, let's keep it to that. They're breeding within the same colony, which means that a set of mice were initially brought about, and then among them, mating has been continued. So in that case, how does the inheritance occur? We'll come to that. Here, the letters small b, designate a mouse which is homologous for the H2B MHC haplotype. If you find this confusing, let's just quickly look at the mouse arrangement. Yeah. So this is the mouse arrangement for, in their case, they have H2 as the locus instead of HLA. And among them, the regions are different. They have H2K, they have H2D, H2L, etc. So in this case, it is just a hypothetical schematic arrangement, and there they are designating a homozygous mouse that is carrying the H2B MHC haplotype. KK is homozygous for the H2K haplotype, and BK, of course, is a heterozygote. Since MHCs are very closely linked and inherited as a set, the MHC haplotype of F1 progeny from the mating of two different inbred strains can be predicted very easily. So what is seen here, coat color is the point that is leading to a difference. So in this case, the coat color is, I mean, it's sort of a grafting or a coat color change experiment that was done. So when they took a B haplotype mouse that is shown here, and they took a K haplotype mouse, and when they were breeding with each other, what they got as the F1 progeny was one which was a heterozygote, that was BK, and that has been indicated in the coat color as well. When they did the other experiment where they tried to actually trace back the way in which inheritance was studied, they performed something different. They tried to do skin transplantation. The question is, why is it done? If you remember, we had talked about HLA compatibility being something that is actively checked in patients before before any sort of transplantation or any sort of grafting is actually done. You have to see that there is compatibility between the MHCs of the individual who is the recipient along with the individual who is the donor for that particular organ. In this case, instead of an organ, what they tried to look at was a skin graft, because it is the difference in coat color, and they try to, coat color is just a schematic representation here. It's not the actual case, but yeah, basically what they try to see is whether if you introduce a skin graft, that is a portion of the skin from one mouse having one particular haplotype into another mouse of a different haplotype, whether or not that skin graft is accepted or rejected. That is what you will eventually be able to understand. So acceptance or rejection of skin grafts is controlled by the MHC type of the inbred mouse. The progeny of the cross between two inbred strains with different MHC haplotypes, again here you have H2B and H2K for reference. The progeny that has both these MHC haplotypes will express BK like we saw previously. And will accept grafts from either parent and from one another, but the parent strain will accept grafts, neither of the parent strains will accept grafts from the offspring. How is it happening? Let's focus on it closely. Here you have the skin graft donor in the center, and you have the progeny recipient, or you have the parental recipient. What are the different types of systems that are being taken into consideration? Either it is a mouse that is homozygous for H2B allele, or it is the homozygous for H2K allele, or it is an F1 progeny, which is a heterozygote, or it carries one of both of the alleles. These are our three participants in the experiment. Let's put it that way. When the skin graft donor is a parent, which is homozygous for the BB allele, which is a BB parent, then when they donate it to the parental recipients, to either BB mouse or KK mouse, only the BB mouse is able to accept it, because there is MHC compatibility or HLA compatibility, or in this case H2B compatibility between these two mice. However, the KK mice will not accept this graft and will remove it. On the other hand, when we have a BK mouse who was a heterozygote carrying both of the alleles, it is able to accept the skin graft from this BB parent. Secondly, when we talk about a KK parent, you see the exact opposite, that only another KK parent is able to accept it, not the BB parent. But on the other hand, a BK heterozygote is able to accept this graft. Thirdly, when it is a BK progeny, that is a heterozygous mouse, its skin graft cannot be accepted by either of the parents. Why not? Because they are expressing, or they have on their surface the alleles for both B and K MHCs. Only their acceptance is possible by the BK progeny. So what do we understand from here? If in case it has been confusing, I'll just sum it up quickly. It only means that when there is expression of two types of alleles on the surface, then only the recipient that carries the MHCs for both these types of alleles will be able to accept it. If there is a MHC incompatibility, then it cannot be inherited. So these are the two main conclusions from here, that first of all, there must be MHC compatibility. Second of all, the inheritance of MHCs is occurring as a whole, that it is occurring because of the linkage disequilibrium, which is leading to its inheritance as a haplotype. And therefore, when the entire BB parent is donating its organ, or whatever, it's donating its skin graft to another BB parent, it is able to sustain the same, but it's not possible in case of the KK parent, but is again possible in case of the progeny, because the progeny has inherited the entire locus. So their compatibility exists. So this is how experimentally people were able to study that there is no recombination which is occurring, and instead inheritance is occurring as a haplotype. I hope it was clear. It's a slightly complicated experiment. If not, please interrupt and let me know. I'll try to explain it better, and I'll try to explain it in a more concise manner. So this is how inheritance was studied and how it was figured out. Then comes the inheritance of HLA haplotypes in a human family. This is just a hypothetical situation. For ease, the human paternal HLA haplotypes are arbitrarily designated as A and B. While the maternal haplotypes have been denoted as C and D. So when the father is donating A and B MHC haplotypes, the mother is donating C and D. These are all the possible progenies that can exist. In most cases, you will not see any recombination, and instead what you will get are either AC, BC, AD, or BD, which are the four progenies that you see. Only in very rare cases, as is indicated in the legend as well, a new haplotype, which is shown as R, can arise from a very, very rare recombination of a parental haplotype. It has just been indicated here because when you talk about biology, you can never say anything with absolute 110% surety that something is completely impossible. You always have to put it in terms of probability, that there is a high probability, much, much high probability of this event occurring, but the reverse or the alternative is always true. So while recombination will not occur in 99% of the cases, there always remains the possibility that by some, some dysregulation, there is recombination, and therefore the child inherits a inherits the haplotype of both parents, but does so in a recombined manner, and therefore has a whole new arrangement of MHC genes. So that is the rare possibility which is shown at the center. In most cases, you will have inheritance as an entire haplotype, and therefore these are the commonly possible HLA haplotypes within a typical human family. So this is what you see here. So we studied the way in which inheritance is taking place in both mice and in humans as well. Now coming to the next part, that how these MHC molecules are expressed on the surface and how it actually is presented to is capable of presenting antigenic peptides. So what you see here on the left is a diagram that illustrates the MHC molecules expressed on antigen presenting cells of a heterozygous mouse. So both the maternal and paternal MHC genes are showing codominant expression. So this is the second major characteristic feature of MHCs. The first was polymorphism. Second was its inheritance as an entire haplotype. Third is its role in skin grafting or any sort of transplantation for that matter. Fourth is the fact that they show codominant expression. What does that mean? If you have studied Mendelian genetics, you're already familiar with it. So there is dominant expression, recessive expression, and also codominant expression. So when there is simultaneous expression of both alleles, um, and one is not overtaking the other one, then there is codominance. Both are equally dominant. There is also another phenomenon known as epistasis. I don't know if you're familiar with it. You can look at it. So epistasis is a different phenomenon. I'll not get into details of it, but there is a one of the alleles shows epistatic behavior, which means it predominates slightly over the other one, but the expression of the other allele is still there. So again, this is a complicated phenomenon. We'll not get there. So this is codominance in this case. And when we talk about codominance, it means that both of the alleles are independently expressed. So in this case, both maternal and paternal alleles will together contribute in the formation of the MHCs that are present finally on the surface. So here representation has been done in the form of colors. If we look at class 1 MHCs, you are seeing DK, LK, and DD and LD for both maternal and paternal MHCs. And what you see on the surface is that there is expression of each of these genes, and they are all forming receptors which are present at the surface. When we talk about paternal MHCs, you are seeing that there is some combinatorial association which is occurring in this case. How so? If you look at the paternal MHCs, you will see some light pink, dark pink, yellow, and orange, orange genes that are present there. So for IE, alpha K, beta K, or alpha D, beta D can either form homodimers or they can form heterodimers and then express on the surface. When they are forming homodimers, it's completely yellow or completely orange. When there is combinatorial association, you are able to see these different association between alpha and beta chains, and that is leading to the formation of different MHCs. So the reason why there should be codominance, or it's rather important in case of MHC expression, is because it is possible that the heterodimer has a higher affinity for the peptide that it is able to present, and therefore is more efficient at doing its job than a homodimer. So in that case, you don't want to miss out on the on better activity of a particular MHC just because it was not expressed on the surface. So therefore, there is codominant expression, and the same also exists for IA as well. So let us go back to the legend again. So because the class 2 molecules are heterodimers, the new molecules containing one maternally derived and one paternally derived chain are also produced. This increases the diversity of MHC class 2 molecules on the cell surface. The 2 microglobulin or beta microglobulin component on class 1 molecules, which is shown here in pink, these side pink associations that you can see. This is encoded by a non-polymorphic gene on a separate chromosome and might be derived from either parent. And it is, when we say it's non-polymorphic, it means that its sequence does not extensively vary within the entire population. Or you can put it in a different way, that it is mostly conserved. So from there, the beta 2 microglobulin gene can be inherited, and therefore it co-associates with the alpha gene, and therefore it forms the final MHC1 receptor. So this is how codominant expression takes place on the surface of antigen presenting cells or APCs. Now coming to the fact that MHCs are very variable. So I had mentioned it before that there is a lot of variability in the alleles that exist across the population. Otherwise, we put it in the form of polymorphism, that there is a large amount of polymorphism that exists in MHCs across the entire population. How does it happen? I'll come to that just a second. So in the external domains, sorry, in the external domains, how can you actually understand the sync between these two? So yeah, this is showing you the entire locus of class 1 HLA molecules for reference. And this is showing you the residue number. So from 0 to approximately, I think, 270 or something, the entire stretch of amino acid residues has been represented in the form of sort of a histogram, where you can see which regions have what extent of variability. So so among that also, alpha 1, alpha 2, and alpha 3 of class 1 HLA molecules are showing you different um, frequencies of variation, which is represented by the graph. In the external domains, most of the variable regions are in membrane distal 1 and distal 2 domains. Location of polymorphic amino acid residues has been shown here in red in the 1 or 2 domain of human class 1 MHC molecule. So this is showing you the extent of variability, and it is represented in this form also in the 3D structure. And there you can see that the part which is actually involved in peptide binding, if you remember, don't think I have that here, but yeah, we can maybe look at the space filling model for reference. So yeah, so if you remember, this is how the peptide binding cleft is formed, right? The central regions, or do I have? Okay, no, I don't have the ribbon structure. But yeah, if you remember, it was there in the lectures. And you can see that the major variations or variability that is present is indicated here in these pink colored stretches. And these are the regions which have maximum variation because they come into contact with the peptide in this region. You can draw this analogy with that of the hypervariable regions or the CDRs of antibodies, which had the maximum variation because that increased the diversity that could be allowed for antibodies to interact with their cognate antigens. So similarly, because this is the region that actually is involved in peptide binding and holding that peptide at a higher affinity, they have maximum variation, or they can, it's necessary for them to have maximum variation. Let's put it that way. So yeah, this was just to show you the extent of diversity across the entire stretch. Uh, this is a screenshot actually from the lecture that Professor Shudeep was discussing, and this I just kept it here so that we can quickly revise the difference between the way in which peptides are processed before we get to more details about it. I'll come to that antigen presentation part soon. But here we are just looking at the type of pathogen and the way in which it is overview of the way in which it is tackled. So first of all, if it is a cytosolic pathogen, if you remember, its degradation will occur in the cytosol, obviously, and these peptides would be able to bind to MHC class 1, and they'll be presented to CD8 cytotoxic cells, and the effect is that the cell which was initially presenting it will die. Second, if it is an intravesicular pathogen, which is going inside macrophages and going inside the vesicles of macrophages and staying there, sustaining there, then in that case, its degradation occurs in endocytic vesicles at a very low pH or an acidic pH. Third is the no, CD4 T cells in comparison to CD8 cytotoxic cells. And the effect on the presenting cell is that it gets activated to kill these bacteria and these parasites. And thirdly, when we talk about extracellular pathogens and toxins, then in that case, obviously, its degradation is also in endocytic vesicles and its presentation is via class 2 MHCs. Effector CD4 T cells or CD4 T helper cells that are effectors will be on the receiving end of the presentation, and the effect is that the presenting cell, the B cell will secrete immunoglobulins to eliminate the extracellular bacteria or toxins. So depending upon whether it's an intracellular or a cytosolic, an intravesicular or an extracellular pathogen, the type of cell that will majorly come in during the presentation and will be involved in the subsequent response is different, and therefore this shows a comparison between any of them. Keep in mind that class 1 MHCs, while they are I think I already said it in the previous class, but class 1 MHCs, while they're expressed by almost every nucleated cell, uh, what remains true is that professional antigen presenting cells, which are capable of actually inducing the T cytotoxic activity, are only of three types, that is dendritic cells, B cells, and macrophages. Whereas any other cell can initially present it, there has to be further cross presentation, and that is taken care of by other cells. So this is again part of something that has been discussed in the lecture, so I'm not getting into that much detail about it, and instead moving on to the initial overview of the endogenous and exogenous pathways for processing antigens. We will be discussing this in more detail in like 3 or 4 slides, but for the initial part, what you need to see is the overview of the process which is followed. Okay, so there are two ways of actually classifying this. What we saw initially was the type of pathogen that comes in. So whether it is cytosolic, intravesicular, or it is extracellular in nature, the way in which it was tackled was different. Here we are addressing it based on the pathway which is involved in antigen processing and presentation. So in the endogenous pathway and exogenous pathway, different factors are coming in. First, we look at the endogenous pathway, which is for class 1 MHCs. Endogenous antigen is present, shown here in green. That endogenous antigen will be chopped up by the proteasome. We'll be coming to that in the next part of this slide itself. And after processing, the peptides that are present are taking it up into the rough endoplasmic reticulum. Following that, within the RER, class 1 MHC is associating with beta 2 microglobulin. Here you can just look at it carefully. This was initially just a class 1 MHC molecule. Then it is associating with this blue colored beta 2 microglobulin. And when this cognate antigenic peptide is present there, class 1 MHC loads it, forms a vesicle, moves from the ER into the Golgi, and from the cis medial and trans Golgi, which are part of the Golgi complex. It's not exactly shown here, but if you have studied the process of vesicular trafficking and vesicular transport, rather, for that matter, previously, you would know that there exists an extensive membranous network inside the cell, and it starts off with obviously the nucleus. From the nucleus, you have directly in association with it the endoplasmic reticulum. From the endoplasmic reticulum, there's a chain of Golgi, and within Golgi, cis, medial, and trans Golgi are present. Cis is the side that faces the nucleus. Medial is obviously in the middle. Trans faces the outside. So it passes through the cis, through the medial, through the trans Golgi, fuses with other or forms another, pinches off as another vesicle, and then that vesicle fuses with the plasma membrane, and then it is presented on the surface or it can be released. So that is how the vesicular transport network works. In this case, what happens here is that the peptides are loaded onto or within a vesicle, then that fuses with the Golgi complex, and from the Golgi complex, it eventually gets presented on the surface. Yeah. Okay. And here in more detail, if we look at it, then what is mentioned in the legend is that in the endogenous pathway, you can see this simple process being followed. But when we come to the exogenous pathway, which is shown here on the right, some things are happening in a different manner. What happens there? In the exogenous pathway shown on the right, extracellular antigens are engulfed into endocytic compartments where they are degraded by acidic pH dependent endosomal and lysosomal enzymes. The antigenic peptides from proteasome cleavage and those from endocytic compartments associate with class 1 or class 2 respectively. So this becomes a major point of distinction between class 1 antigens and class 2 antigens. For class 1 antigens, the antigenic peptides are generated here from the proteasome. It is much ahead. It is occurring at the RER, or in fact prior to the RER. Whereas for exogenous pathogens, exogenous antigens, it's different. And yeah, and what you see there is that there is a vesicle. Within that vesicle, the pH change that occurs, which is shift towards an acidic pH, is what activates the enzymes that are present within that autophagosome, and then that undergoes the process of digesting those peptides, loading it onto MHC class 2, and then presenting it on the surface. The antigenic peptides are presented or are transported to the cell membrane, and it should be noted that the ultimate fate of most peptides inside a cell is neither of these two pathways, but rather to be completely degraded into amino acids. Why is this important? This is because if your system had started processing every single protein molecule that exists within the cell, then that would lead to a huge wastage of time and huge wastage of resources. How so? For example, if you consume something, some sort of food item, and instead of that becoming a source of nutrition for your body, instead if it is going into activation of the immune system, then that would trigger a non-specific and very dangerous response. Secondly, it would not justify the purpose of either the immune system or of the food item, right? So in that case, most of the antigens, while anything that is not immediately recognized as a threat or as a pathogen, does not immediately go into antigen processing. Instead, what it is undergoing is degradation to form small amino acids, which will replenish the amino acid pool of the system and will add to the amino acid pool of the system, which will allow more and more protein synthesis. And that will obviously be an essential nutrient in the diet of most individuals, and therefore it's required, not diet only, for the functioning of an individual, and therefore it is required. So these are not the default pathways. This is only activated in case of an antigen that has come from a pathogen and that needs to be eliminated as soon as possible. So this is the difference between those two pathways, that what triggers the formation of those antigenic peptides. On the right, what you see is the proteolytic degradation system for intracellular proteins or endogenous antigens. So endogenous proteins may be targeted for degradation by ubiquitin conjugation. So these proteins are degraded by the 26S proteasomal complex. So this was already, I think, explained really well by Professor Shudeep, how the proteasome actually looks like. It has these sort of this cap and bottom region, which is the 19S regulator, and a central sort of uh, pipe-like structure, or rather what he described as a channel. Yeah. So what he described as a channel, or the region which actually interacts with the peptide, and that is the 20S proteasome. So this is your protein molecule or the endogenous antigen of interest that first is recognized as something that needs to be eliminated from this system. And for that purpose, it is actually chopped up, and that chopping up is taken care of by the proteasome. So to allow the proteasome to even act upon this particular peptide, there is one step that must occur in prior, and that is polyubiquitinylation, or otherwise known as ubiquitin conjugation. When we say polyubiquitinylation, it means multiple ubiquitin moieties are added. Moiety simply means entity or group. I think I had described it before as well. So initially there's only just one tag, and in that case, ubiquitin can also be sort of a regular modification that is present for the normal functioning of proteins. But when there are multiple ubiquitins that are one after each other in tandem, then that is identified as an eat me signal, which means that the cell is indicating that this particular protein needs to be removed as soon as possible. So in that case, polyubiquitination has a role to play, and then that protein will be brought to the proteasome. And here you see the constitutive versus immunoproteasome and how they are present. What is the difference between those two? Let's find out from the legend. So cytosolic proteolytic system for degradation of intracellular proteins has been indicated here. First, the endogenous proteins may be targeted for degradation by ubiquitin conjugation. These proteins are degraded by this complex, the 26S proteasome complex, including the 20S constitutive proteasome and a 19S regulator. However, in section B, what you see is the is for activated APCs or antigen presenting cells that you are able to see that there are, instead of constitutive proteasome, you're able to see immunoproteasomes. Where are they coming from? They're coming from the LMP genes. If you remember, we had seen LMP genes here. Yeah, so we had seen all these LMP genes that are present here, and they are contributing towards the formation of the immunoproteasome. And these immunoproteasomes, they have certain residues which have been replaced, or certain proteins which have been replaced, and they have some specific role to play. And therefore, this immunoproteasome is more efficient for creating peptides that can assemble with MHC class 1 molecules, and therefore that is its job, and that is expressed by the professional antigen presenting cells. So this is how the proteasome is extremely crucial and is very, very functional in doing its job. uh, Next comes the role of tapacin. So what have we brushed up till now? The different properties of MHCs, how they exactly work, the fact that they are polymorphic, that their inheritance is as a haplotype, the fact that you can study that inheritance using a very simple mice grafting experiment, the fact that its inheritance is codominant in nature, that is, both the alleles are expressed and are expressed almost equally, where that variability exactly lies, what are the ways in which different subtypes of pathogens are tackled by the immune system, and um, what are the cells that act upon it, and what is the long-term effect of this immune response. All of that we looked at. And we also saw the key differences between exogenous and endogenous antigen processing pathways, and how in one case, or in the case of MHC1, it is actually the proteasome which takes over this job. Excuse me. In case of MHC2, it is actually the pH of the phagosome which activates the enzymes, and that chews up the peptide to eventually lead to removal of that not removal, let's say it's just the chopping up of the peptide for its presentation. And the role of the immunoproteasome and how that is encoded. Now we come to the next part, which is that after the peptide has been generated, what exactly is facilitating its loading on the surface. So here you see TAP, the role of TAP, which is transporter associated with antigen processing. It's also called as tapacin. So TAP is assembled because of a heterodimeric association between TAP1 and TAP2. Again, TAP1 and TAP2 were also encoded within the MHC locus itself. So yeah, TAP1, TAP2 are these two elements. Here you can see TAP1 in black, here you can see TAP2 in black within the MHC2 locus. So again, it's not necessary that if its job is majorly taken care of by MHC1, then it cannot be present within the MHC2 locus. It's not that hard and fast. They are all located very close to each other, so that their inheritance is not overall affected. But yeah, the subtle differences can persist. So this shows the schematic diagram of TAP, which is a heterodimer anchored in the membrane of the rough endoplasmic reticulum, or RER. The two chains are encoded by TAP1 and TAP2 gene segments. The cytosolic domain in each TAP subunit contains an ATP binding site, and the peptide transport depends on the hydrolysis of ATP. So TAP subunit is it's an active transporter. Or what is the reason we are specifically seeing it? That is because transporters can either be active, passive, or they're able to act as co-transporters. For example, sodium glucose co-transporter, where on one hand it is, it is taking, okay, so here there are multiple ways of classifying transporters as well. Either it can be classified based on whether it is energy dependent or not, or a second brand of classification can be done based on if it is able to transport, for co-transporters, if it is able to transport it in the same or in opposite directions. So based on the energy factor, there are active transporters and passive transporters, or facultative or facilitative transportation can also occur, or co-transportation can also occur. And under co-transport, you have symport, antiport, etc. Also, symport means uh, entities are transferred into the same direction. Antiport means they're in opposite direction. Say for example, if you have heard of the okay, Na plus K plus transporter, which has a huge role to play in maintaining action potential for conveying any sort of information between two neurons. So in that case, Na plus and K plus are moved in completely opposite directions. So Na plus, 3 Na plus out and 2 Na plus in is how it works across the membrane. So across the plasma membrane, the movement of Na plus and K plus are in opposite directions. So that is an antiport transporter, but it does not always have to be energy linked. There are in some cases no association of energy, and then in that case, that is it's not active transport in those cases. In case of Tapacin, however, there is a huge role of ATP. It is ATP dependent, and therefore its action is energy intensive. So yeah, peptide transport is very heavily dependent upon the energy that becomes available or accessible upon ATP hydrolysis. In the cytosol, what is shown in figure B, in the cytosol, the association of these colored spheres that you saw with the proteasome changes its catalytic specificity. How so? So from the constitutive proteasome, the immunoproteasome has some difference in its catalytic specificity, and it favors the production of antigenic peptides that are able to bind to class 1 MHC molecules. Then these peptides are translocated by TAP into the RER lumen, rough endoplasmic reticulum lumen, and in a process mediated by several other proteins, they eventually associate with class 1 MHC molecules, and then they are able to then they are presented at the cell surface. So what you see here is that this main job of moving peptides into the rough endoplasmic reticulum from the cytosol is taken care of hugely by this TAP system, or by the TAP proteins or the Tapacin proteins, TAP1 and TAP2, in an energy dependent manner. When TAP is not functional, then what happens? So one example that you see here is of a disease which is called as necrotizing granulomatous. It's TAP deficiency syndrome, and it leads to formation of necrotizing granulomatous lesions. What does that mean? Necrotizing means tissue that is dying or that undergoes necrosis, and lesions are these sort of scabs on the surface or these injuries on the surface, and they are granulomatous in nature, and they are at the midface of a patient that has this TAP deficiency syndrome. So TAP deficiency leads to a condition with symptoms characteristic of autoimmunity, such as these skin lesions, as is shown here, that appear on the extremities and in the midface, as well as immunodeficiency that cause chronic sinusitis leading to recurrent lung infections. This is just a representative of a disease, because I wanted you to understand the significance of Tapacin. In today's time, Tapacin is studied extensively, and is a very it's of great interest in immunotherapy as well. You can read about it. It's a very interesting topic. But yeah, you understand that simply just keep this in mind, that MHC class 1 is what eventually triggers the T cytotoxic cell response. It is presenting antigens. Cells with MHC class 1, specifically the professional antigen presenting cells, are presenting the antigenic peptides to cytotoxic T cells, which will kill and remove it from the system. So it's doing a hugely important job. Now, MHC class 1 is, again, if we reverse it a little bit, very, very hugely dependent upon Tapacin. Because if Tapacin does not work properly, or if TAP does not work properly, it will not allow the entry, or will not facilitate the entry of peptides from the cytosol into the RER lumen. That peptide can never be presented if it does not even enter the RER lumen, and therefore that person will have a severely compromised immune system. So that is what you see here, that they keep getting lesions and, they suffer from a lot of pain. It's obviously a horrible disease, but yeah, it's not very abundant. It is certainly rare. Let's keep that in mind. But yeah, it does occur. It's not an impossible uh, occurrence. So yeah, that's how I wanted to highlight how TAP works and why it is so relevant and what can be its far-reaching consequences of TAP not working correctly. So yeah, that was the job of TAP. Now comes the question of how exactly does this assembly and stabilization occur. So when we talk about class 1 MHC molecules, what are the different substeps? Till here, what we saw is that first the proteasome is coming in, chopping the antigen. Second, we saw that after the antigen has been chopped by the proteasome, specifically the immunoproteasome carrying certain residues which are different from the regular or constitutive proteasome. And from there, the peptides that have been generated can either lead to production of amino acids, or some part of them can be loaded by TAP onto the surface, and then that onto the RER, or within the RER, and from the RER it will form vesicles that go through cis, medial, trans Golgi, finally to the surface. Now what happens after the RER is being elaborated here. Within the RER, or rough endoplasmic reticulum, we see that a newly synthesized class 1 chain associates first with calnexin. So this is your class 1 MHC alpha chain that is associating with the molecular chaperone known as calnexin, following which it also associates with this orange color cap-like structure known as ERP57. So let's come to the legend and then we'll go into details about the figure. So within the rough endoplasmic membrane, a newly synthesized class 1 MHC chain associates with calnexin, a molecular chaperone, and ERP57 until beta 2 microglobulin binds to the chain. So it does so up till until beta 2 microglobulin binds. So why is this necessary? It's necessary to keep the class 1 MHC stable until it has the association of beta 2 microglobulin. Before that, the structure is not stable at all. So once beta 2 microglobulin comes in, you can see the simultaneous removal of calnexin almost at the same time. Furthermore, there is also association of tapacin, and there is also association of calreticulin in this case. So tapacin associates very closely with class 1 MHC, and calreticulin tapacin associated class 1 MHC molecule is what is actually seen here. So what happens on the other hand from the cytosol? The peptide comes chopped up by proteasome, generates antigenic peptides, and then that antigenic peptides move from the cytosol via tapacin into the RER lumen. Within the RER lumen, it can be removed by ERAP, which is a peptidase, if I'm not wrong. Let's just go back to the legend again. Yeah, the binding of beta 2 microglobulin releases the calnexin and allows binding to calreticulin and to tapacin, which is associated with the peptide transporter TAP. This associate promotes the binding of an antigenic peptide, and these antigens can be further processed by exopeptidases such as ERAP1, producing some fragments that are most suited for binding to class 1 MHC. This association stabilizes class 1 molecule peptide complex, allowing it to be transported from the RER to the plasma membrane. So what you see at the end is that once peptides have moved into the lumen via the TAP proteins, it can then associate with the calreticulin tapacin associated class 1 MHC molecule, and then that all of the other chaperones that dissociate, and this is a loaded class 1 MHC, which will then exit the RER, form more vesicles, move towards the cell surface, and present its antigen to the cytotoxic T cells. So these are the substeps, the other chaperones that participate in this process, and how crucial they are, because without them, the MHC integrity cannot be maintained. So yeah, this is for class 1 MHC. On the other hand, for exogenous pathogens or exogenous peptides, let's say, for the generation of antigenic peptides in the exogenous processing pathway, what you see on the right is that the internalized exogenous antigen moves through several acidic compartments, finally ending in the specialized late endosome, specialized MHC late endosomes, where it becomes degraded into peptide fragments, which are associated with class 2 MHC molecules transported in vesicles from the Golgi complex. Lot of information. Let's go over it. So what you see first is the antigen. That antigen is being internalized by several types of processes, sometimes called as phagocytosis, sometimes it is called as pinocytosis also. So in this case, here you have clathrin coated vesicles. No need to get into details if you're not already familiar with it. But this vesicle structure formation is dependent upon um, some molecular level processes. I'll not get into details of it, but clathrin is involved in it. So think of it as how you have a dough. So from that dough, when you make a little, for example, for if you're making say roti, from the entire dough you will take out little snippets, and that is how vesicular pinching off occurs. It's the easiest mechanism I could think of, is that you sort of twist and pinch off a portion. So that's what happens, is that from the membrane a portion of it sort of internalizes and then pinches off, and the rest of the membrane can again dissociate. Why is it able to do so? Is because it's a fluid mosaic bilayer, a lipid bilayer, and therefore nothing is very rigid there. Of course, there are certain regions which are somewhat rigid, but it's quite fluid in nature, and therefore a portion of it, even if it is removed, the rest of it can again join back together and make an intact membrane. So this vesicle that has pinched off carries your antigen with the antibody, for example, as shown in the figure. It carries that, and then that passes through several stages where there's an early endosome with a pH of approximately 6 to 6.5, and from there it has a it also has a late endosome where there's a pH of 4.5 to 5, and there you can see that these individual antigenic peptides that are present within that endosome because of the acidic pH will get loaded onto MHC class 2. So where does MHC class 2 come from? From the Golgi complex. MHC class 2 is also released in a vesicle. It also carries something known as the invariant chain. If you have already listened to Professor Shudeep's lecture, you would know that invariant chain helps keep the MHC structure stable until or till the point that antigen can be loaded onto it. So this MHC class 2 along with the invariant chain is present. So you have two sides. From one end, the MHC is coming. From the other end, the antigenic peptide is coming. Both the vesicles are fusing with each other within the late endosome. Their loading is occurring, and after loading has been facilitated, such a vesicle will move towards the surface and will express or will present the antigen which is already loaded on MHC class 2. In this particular schematic, the cell that is shown here is a B cell, which internalizes antigen by receptor-mediated endocytosis with the membrane-bound antibody functioning as an antigen-specific receptor. So the receptor is at the surface, it is receiving its ligand, and then it is endocytosing. And upon endocytosis, it is moving towards, I said phagocytosis before, that was not correct. I was thinking of endocytosis. So yeah, it endocytoses that particular antigen and then fuses with a vesicle that carries MHC class 2. Then it allows the loading of the peptide that occurs in a sort of trial and error manner. Whichever is the most efficient is able to stay strongly associated or is able to stay with very high affinity with MHC class 2, and then it can be used to present on the surface. So yeah, that's how it works. I gave a lot of information. If there is doubt, please do interrupt me. Okay, so we are at the last slide. It won't take very long. If anyone has any doubts, please go ahead and ask because there was a lot of information that I gave today. So finally, during this assembly, what are the more subtle details that are occurring? First of all, the fact that MHC class 2 molecules, how are they assembling? First, you had this MHC class 2, which carried the alpha and beta chains. With that, you have an invariant chain that is associating and forming this structure. Associated with that is the clip, which you already are familiar with from professor's lecture, that initially there is a longer peptide that is membrane bound, then both of these sides are chopped off, and then what remains at the center of MHC class 2 is the clip, invariant chain clip. And the smaller fragment remains bound to this groove of HLA or class 2 MHC molecule. Then what happens? That there is association of HLA-DM, which comes in, which is a non-classical MHC class 2 molecule present within this compartment, which mediates the exchange of antigenic peptides for CLIP. So it binds, it allows the binding of an antigenic peptide. When the antigenic peptide is strong enough, it then dissociates and allows the release of CLIP to occur. The non-classical class 2 MHC HLA-DO may act as a negative regulator of this processing by binding to HLA-DM and inhibiting its role in the dissociation of CLIP from class 2 MHC molecule. So both HLA-DO and DM are non-classical MHCs which help in this process of dissociating CLIP and association of antigenic peptides. So finally, what happens is that you have released CLIP, you have a class 2 MHC molecule that is bound to a peptide, then that can be loaded onto the surface. So this entire process is taking place within a vesicular structure which has an acidic pH, where all these enzymes are able to do their job. They're able to chop off what needs to be chopped off. They have all these, say, hundreds of options of antigenic peptides which can be loaded and unloaded continuously until they find a perfect match where it has very high affinity and can present the antigen on the surface for an extended period of time. So this is how antigen presentation for class 2 MHC molecules actually works. So with all this information that we have until now, let's sum up the entire process. First, let's look at the endogenous pathway via class 1 MHCs. So you have an endogenous antigen. That endogenous antigen gets degraded by the proteasome. What sort of proteasomes? These are called as immunoproteasomes, which, if you remember, carry certain residues which alter the specificity of the proteasome in such a way that it prefers to bind to class 1, prefers to bind to those peptides which can digest class 1 antigens. So that is an immunoproteasome. Then, once these peptides have been generated, these peptides are loaded and transferred by TAP, which takes over this job, which is the transporter of antigenic peptides, and it moves all of these into the rough endoplasmic reticulum. Once they are present within the rough endoplasmic reticulum, multiple chaperones come in. The first chaperone that comes in is calnexin, then comes ERP57, then comes tapacin. All of these associate together. And following this, class 1 alpha chain binds to calnexin and ERP57, then beta 2 microglobulin. Once beta 2 microglobulin has bound, calnexin will dissociate, calreticulin and tapacin bind uh, to each other, and then MHC captures the peptides and all the chaperones will fall off once the peptide has been loaded onto MHC class 1. Finally, once it has been loaded, this particular peptide will pinch off from the rough endoplasmic reticulum, will fuse through the Golgi, and finally, this is transported from the RER to the Golgi complex to the plasma membrane, and then finally expressed on the surface. So this is the entire process of the endogenous pathway. On the other hand, when it is an exogenous antigen, then in that case, class 2 MHCs come in. First of all, the class 2 MHC alpha and MHC beta molecules, they bind to invariant chains, blocking the binding of the endogenous antigen. Secondly, MHC complex is routed through the Golgi to the endocytic pathway compartments. Why? Because interaction does not occur within the RER. Interaction does not occur within the rough endoplasmic reticulum, but instead occurs within the autophagosome, as we discussed. So in that case, just the class 2 MHC molecule bound to the invariant chain is moving out of the rough endoplasmic reticulum, going through the Golgi, and from the Golgi it is associating with the CLIP protease, and then that CLIP protease is removing that, not CLIP protease, it is actually allowing CLIP to persist while the rest of the peptide is digested. And then this CLIP containing MHC2 interacts with the exogenous antigenic peptides. So here you find the entry of the exogenous antigen being digested into peptides. They interact with each other. Here HLA-DO and DM also come in, and they allow this trial and error method of binding and antigen, checking whether it is efficiently binding with high affinity or not. If not, it should be removed. So that trial and error is carried out. And then finally, the exogenous antigen with antigenic peptide is loaded onto class 2 MHC. And then that class 2 MHC peptide is transported to the plasma membrane, and it can then express the peptide on the surface. And then it will interact with TH cells, and it will either trigger more B cell proliferation or will trigger the appropriate response in respect or with respect to the exogenous peptide or pathogen that has come in. So this is the entire process, the way in which at a molecular level these sub-steps are regulated. So with this, I am done with my slides for today. If there are any doubts, please go ahead and ask. I will also show you some sample questions just for reference. If you have any questions from the parts that have been discussed till now, you please go ahead and ask me because I shared a lot of information. Any doubt from any part, please go ahead and ask me now. I'll open that PDF in the meantime. Okay, if not, then I'm assuming that you were able to understand everything. Uh, Please interact in these sessions. Otherwise, it's very difficult for me to be assured whether you've understood what you were trying to discuss. If any doubts are there, please don't hesitate. Refer to the textbooks. They're very, very helpful. So these were some previous year questions. So just for your reference, the first question is about T cells, that a T cell recognizes an antigen as a peptide, as in the peptide bound form by a particular allelic variant of an MHC molecule, and will not recognize the same peptide bound to other MHC molecules. This behavior of T cells is called as MHC restriction, polymorphism, polygeny, or none. Okay, so I did not discuss this directly in this interaction session, but if you remember, it was there in the first lecture for this week, where that MHC restriction is the condition where peptides that are bound to one particular type of MHC will only be recognizable to a particular T cell and not to others. So that behavior of T cells is called as MHC restriction. Polymorphism and polygeny are of course different. We discussed that extensively in today's session. So the correct answer in this case is MHC restriction. If you want to read up a little bit more on the phenomenon, you can refer to either the lectures or the book itself. I did not have it in my slides, but yeah, you can refer to Kobe or Genbei for that matter. Second question was about the diversity of MHC molecules seen both within an individual and in the population is due to which of the following? Is it due to just a little bit? Yeah. Diversity of MHC molecules seen within an individual and in the population is due to polymorphism only, polygeny only, combination of polymorphy and just combination of polymorphy and polygeny, or because of VBJ recombination. So if you think about polymorphism, it means that within the population, there is diversity of MHC molecules. When you talk about polygeny, it is because a single individual is inheriting MHCs from both their parents. So they have multiple genes which are encoding for the same MHC locus. So you have multiple genes or multiple alleles for the same final protein or same locus. So both polymorphism and polygeny are relevant here. When we talk about an individual, it is polygeny. When we talk about the population, it is polymorphism. Therefore, the combination of the two results in the final diversity of MHC molecules. In this case, VBJ recombination is obviously not relevant. It is for BCRs and TCRs. For MHCs, the arrangement, the genetic loci, everything is completely different. So the correct answer is option C. Then in question 3, we were asked which of the following is correct with respect to T cell activation. So super antigens. Okay, this is also something I did not discuss. Bacterial super antigens and viral super antigens. Again, these were topics that were well detailed discussions of which were carried by Professor Sudeep. So I did not get into it. But super antigens, whether they need to be processed by the endogenous pathway, whether they need to be processed by the exogenous pathway, or do they not require any processing, or whether they can be processed by either of the two. So in this case, if you remember, for T cell activation, superantigens do not need to be processed by exogenous or endogenous pathway. They do not require any processing at all. And that would be the correct answer in this case. If you want, I can include a slide on it maybe in the next session, but I don't have one at hand at this moment. But yeah, bacterial and viral superantigens were well described by Professor Sudeep. So you can refer to that lecture in case you have some confusion regarding it. Then in question 4, the recognition of a target cell by CD8 cytotoxic cells is MHC dependent, MHC1 restricted, MHC2 restricted, both MHC1 and MHC2 restricted. So if you remember, we had very initially when we were comparing the difference between MHC class 1 and class 2, we saw that for CD8 cytotoxic cells, they can only recognize the antigens which are presented by class 1 MHC. And in fact, if we get a little more specific, class 1 MHCs which are present on professional antigen presenting cells can only activate CD8 cytotoxic T cells. So they are only class 1 MHC restricted. And if there was another option to be more specific, class 1 MHCs that are presented by or that are present on the surface of professional antigen presenting cells or professional APCs are only able to activate CD8 cytotoxic T cells, while they can be presented by almost every nucleated cell. So they are MHC class 1 restricted, and that is the correct option. So the way in which you frame the question will can be different, but it is addressing the same topic. On one hand, you can say that cells expressing class 1 MHC are activating, or the effector cells for class 1 MHCs are CD8 cytotoxic T cells, or you can reverse the question and you can speak from the perspective of the CD8 cytotoxic T cells, that they are only able to see or recognize antigens which are presented by class 1 MHC. So they are restricted in that front. So it's the same idea being framed in different manners. So you just need to be a little thorough when you are studying, and it's quite easy to understand and very fascinating, as you already know. So class 1 MHC restricted. The generation of antigenic peptide for MHC class 1 occurs in rough endoplasmic reticulum, cytosol, Golgi apparatus, endosome. So keep again in mind, we have just discussed this in a lot of detail, that antigenic peptide processing for MHC 1 and 2 is very different. For MHC 2, we talked about it entirely occurring within endosomes, but for MHC 1, it is occurring via the immunoproteasomes, and proteasomal processing occurs within the cytosol, and its loading into the RER is taken care of by TAP. And after TAP has facilitated its entry into the RER, only then can its loading occur onto the MHC class 1. So this question it directly is asking you for the generation of antigenic peptide, and that is taken care of by the immunoproteasome within the cytosol. So the cytosol is the correct answer in this case. Then we're asked to choose the correct combination, or basically it's a match the following sort of a question. So you have multiple options. Sorry. You have multiple options here. Just you need to find the correct combination between them. So one thing you can do always is to start in these cases where you have lots of options to address it a little quickly. You can start from the options directly, see which of them are completely different from all of the others, and you start by addressing those. So for example, you'll see that this is common in a lot of cases. 3A is common in B, C, and D. You can automatically assume that it would certainly be true, and then you can verify that first of all, or you can start from the question directly. I'm just trying to give you an idea that if you want to solve a question in a quicker time period, if you have a time-intensive examination at hand, that's how you can try to attempt questions. But right now we are just going over the concepts. So in this case, we are trying to match these. So for for this question, the correct option is option B. So we'll address it directly in that front. That option 1, which is for TAP, it is a transporter of antigenic peptides. So it moves peptides into the ER. Therefore, option B is correct. So 1 is linked with B. Secondly, you have Tapacin. What does Tapacin do? Again, don't get confused between TAP and Tapacin. TAP is the transporter protein which is present across the membrane, and it is allowing the movement of antigenic peptides from the cytosol into the rough endoplasmic reticulum. Tapacin is a molecular chaperone. It is associating with class 1 MHC and maintaining its integrity along with multiple other chaperones. So Tapacin in this case, option 2, associates with D, which is that it forms a bridge between the TAP1, between the class 1 MHC molecule and the TAP complex. So it forms a bridge between the two. It was not there in our schematic, but it was present in Professor Shudeep's lecture. So again, if you want to need to revise that, please go back and look into it once again. But yeah, it forms that bridge between these two and allows the stabilization of MHC class 1 molecule. So 2 associates with D. Then comes option 3, which is ERP57. It breaks and reforms disulfide bonds in the class 1 MHC domain during peptide loading. Now, we did not directly look into it in this case, like in the schematic or in what Professor Shudeep had taught, but we just studied it as a sort of molecular chaperone, that it is involved in some sort of a in folding, refolding, or some sort of protein integrity maintenance activity it is involved. But in this case, we are learning about it in more detail, that it is helping in the formation and removal of disulfide bonds in the class 1 MHC alpha domain during the process of peptide loading. And then finally, calnexin retains the class 1 MHC molecule alpha chain in a partially folded state, and it maintains the integrity of that protein in that case. So in this case, the correct option is option B. So these are the correct matches that you can find in this case. For the next question, antigen binding with MHC2 molecules is mediated by cathepsin S, CLIP, HLA-DM, or peptidyl transferase. So MHC2 molecule binding, if you remember, was facilitated by HLA-DM and HLA-DO. We had that schematic to represent that. HLA-DO was involved as a negative regulator for this process, and it was controlling whether or not HLA-DM can bind, and it can facilitate the loading of peptides, antigenic peptides, onto the MHC molecule. Just for reference, I'll open the PPT for a second. Yeah, so here in this step, you saw that HLA-DO and HLA-DM are involved in this peptide loading process. So that is where their job came in. And therefore, the correct answer in this case would be HLA-DM. Then 3 major cell types, dendritic cells, macrophages, and B cells, present peptides that are bound to MHC class 2 molecules for recognition by CD4 T cells, that is CD4 T helper cells. In general, these peptides are derived from proteins or pathogens taken up by the cell by endocytosis, phagocytosis, or macropinocytosis. Based on these pathways of antigen uptake, some of the enzymes that degrade proteins to generate peptides for MHC class 2 presentation are A. Ubiquitin ligases that tag protein for degradation by the proteasome, ATP transporter proteins that deliver endocytic proteins into the cytosol for degradation, cysteine proteases like cathepsins that function at acidic pH, the lysosomal thiol reductase that is found in endosomes, and lysosome-associated membrane trafficking protein, LAMP2. So these are all of the different options that are there. So in this case, what you need to keep in mind is that the initial part is not very relevant to our question. You directly start the question from this portion where you have been asked based on the pathways, based on the existing pathways for antigen uptake, the enzymes which are functional in protein degradation to generate peptides for class 2 MHC presentation are which of the following? So if you remember clearly, it is actually for MHC class 2, peptide generation is actually occurring within the endosome. And these endosomes, initially, when they're immature, do not allow peptide formation or antigenic chopping up of the antigens that occurs at a specific pH, which is acidic in nature in the late MHC endosomes. So it is the acidic pH which activates certain enzymes which are present within those endosomes and allows the generation of peptides. So based on just this information, the direct option that you can come to is option C, that it is cysteine proteases like cathepsins that function at acidic pH. And this is particularly important because cathepsin, as you can see, it is a protease. If you don't have any knowledge about what has been asked here, you just need to know the nomenclature of enzymes. You'll see multiple types of enzymes that have been mentioned here. There is ligase, protease, reductase, etc. Ligase is something which links something to something else. So ubiquitin ligases, they attach or they tag proteins with ubiquitin. And what does that do? It identifies that protein as something that needs to be degraded. It is a ligase. Ligase as an entity is like a glue. It's a glue that is attaching some particular moiety onto something else. DNA ligase attaches DNA ends with each other. In this case, it is ubiquitin ligase. So there are different types of ligases that are present within the system. That is obviously not going to chop up a protein or not going to generate antigenic peptides. Second, you have transporter proteins. Obviously, they are involved in movement of something or transport of something from one side to another. In this case, it is from into the cytosol for some degradation. So that is not operational here, especially not for class 2 MHC, since we know that everything occurs within an endosome. The third option is for proteases. So that in itself is an answer for you, because protease, as the name indicates, is something which degrades proteins or chops up proteins. In this case, even if you don't know the name of the enzyme, you can figure out that it is a protein degrading moiety, right? So it is functional also at acidic pH. So both these options are correct, because we did not directly talk about cathepsins. That's why I'm telling you how to answer such a question. So you have acidic pH and you have proteases, which are hints that will tell you that this is how you need to answer your question, and this can chop up proteins for MHC class 2 presentation. Thiol reductase is not really necessary here, because especially because it's lysosome associated, which we are not going to directly here. Of course, lysosomes have a role to play here, but it's not operational in this case. And LAMP2 is also not involved here because it is involved in membrane trafficking. So the correct option in this case is option C. So that is how you would address the question. The initial part was not very relevant here. What you directly need to get to is MHC class 2 presentation. So I hope it was clear. If there are any doubts, please interrupt. Uh, then for the next question, during class 1 MHC, during MHC class 1 synthesis and folding in the endoplasmic reticulum, a process of peptide editing takes place as the newly synthesized MHC class 1 protein is held in a peptide receptive state by binding to calreticulin ERP57 tapasin complex. You remember that complex image that we saw. Peptide editing ensures that MHC class 1 molecules that reach the cell surface have stable, high affinity binding for the peptide cargo. It is important to the immune response. Why? A, because it maintains high levels of surface MHC class 1 expression. Do you think that is very relevant? I don't think so, because A, MHC class 1, high availability of just MHC class 1 expression on the surface is not relevant. You need to just have a peptide with very high affinity present at the surface so that it can be identified by the CD8 cytotoxic T cells, which can remove it in due time. Second, it ensures that MHC class 1 molecules are not degraded in the ER. So their degradation is not dependent in this case on peptide editing. It is, if you remember, degradation was controlled by something else. You can go back to the lectures in that case. Here it is not talking about the peptide. Here it is talking about the fact that MHC stability is affected by it. Third is that it retains the nascent MHC class 1 molecule in a peptide receptive state. Fourth, it allows the surface MHC class 1 molecules to bind new peptides from the extracellular milieu. That will obviously not happen. The peptide loading always occurs inside the cell. MHCs cannot bind to peptides directly from the outside. The peptide loading is very specific, very tightly controlled, and it's very important to have that tight control so that most efficient peptide presentation can occur. Finally, option E, which is correct, is that it prevents surface MHC class 1 molecules from undergoing peptide exchange at the cell surface. Why is that relevant? It is because in the rare scenario that there is some other peptide which has much higher affinity for one particular MHC on the surface, what this particular MHC can do in the rare scenario is that it can exchange it with the other peptide and bind to the much stronger peptide which is present at the surface. That would be non-specific, and that is extremely incorrect and should not be allowed by the cell. So instead, what is done is that peptide editing is carried out within the ER at the time of loading to ensure that the peptide having maximum affinity for the MHC is only allowed to be retained, and only then it is expressed at the surface. So option E is correct. Finally, for the last question, this is not really related, but yeah, for the correct statement about delta gamma T cell receptor, they recognize antigen as peptides generated by MHC molecules. They recognize their target antigens directly. They are not restricted by the classical MHC class 1 and class 2 molecules, both B and C or both A and C. So delta gamma T cell receptor, if you remember, TCRs were of two types. Two types or were present in two combinations. We looked into that before, like a few weeks before, 2-3 weeks before. But yeah, they cannot recognize the antigens as peptides presented by MHC molecules. So option A is not correct. They can recognize their target antigens directly. That is the correct option. Option B. But option C is also correct. They are not restricted by classical MHC 1 and 2 molecules. So both statement B and C are correct. As a result, the correct option is option D, that both B and C are the true statements. So with this, yeah, I think I come to the end of my presentation for today. It is the time for questions. If you have any questions, please go ahead and ask. I'll try to address them, or I'll come up with slides in the next session to better address them. Please let me know if you have any doubts. Okay, there are no doubts. Okay, if not, then thank you so much for joining. But please try to be more interactive. I've been asking that for more interaction in all of these sessions. But it's okay. If you have any doubts, you can reserve them for the final session that we'll have at the end of the course. So after 12 weeks, there will be another extra session that we'll have where we'll just take up questions from the entire course, all of the lectures that have been covered. We'll go through some questions. I'll come up with some. We'll discuss the assignments if needed. And if you have any doubts, you can note them up, any topic that you need more clarity on, and we'll address that thing. So thank you so much for joining. I'll end the session for today. Have a good evening. Thank you.